Welcome, everyone, to the Office of the Chief Scientist Seminar Series. My name is Andrew Paul. I'm a Senior Science Advisor with the OCS, and I will be your host for the fall set of seminars this year. I'd just like to start by acknowledging that Alberta is on lands and gathering places for Treaty 4, Treaty 6, Treaty 7, Treaty 8, and Treaty 10, as well as homeland of the Métis. This includes traditional territory of the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, and Saulteaux nations, whose history, language, culture, and governance have shaped the province that we are all privileged to share. Wherever you find yourself today, I encourage you to recognize and acknowledge the relationships with First Nations, Inuit, Métis, and the land upon which you may live, work, and learn upon. Alberta's Chief Scientist provides these seminars as an opportunity to exchange knowledge on interesting sciences happening throughout Alberta in an open and sharing environment. The intent of these seminars is to build awareness and understanding on different topics and to put that knowledge into practice through the use of evidence-informed decision-making. Today, your video and audio has been disabled to maintain our call quality, but please enter any questions or comments you might have during today's presentation using the chat window in Teams. Once today's presentation is concluded, the presentation portion is concluded, I'll read out these questions in the order that they were received, and our speaker can respond to them. And with that, I would like to introduce today's speaker, and that is Delaney Frame, who will be presenting on social interactions of cougars in West Central Alberta. Delaney has over 20 years experience working for government within both lands and wildlife departments, and currently works as a senior wildlife biologist with Alberta Environment and Protected Areas. Delaney spent 5 years as a field team member of the Cougar Adaptive Management Project, which led to her Master's of Science examining behavior cougars in Alberta. And with that, Delaney, I'll turn things over to you.
Perfect. Hi, everyone, and thanks, Andy, for the introduction. And thanks, everybody who logged on to hear this talk today. I had the pleasure of working on this project for 5 years, and I'm always excited to talk about some of the things that we've learned. Okay, now it's working. There we go. So cougars are habitat generalists, making them among the most adaptable and wide-ranging of mammals in the world. So their range extends, as you can see on this map, from northern Alberta and British Columbia to the southern tip of South America. In Alberta, cougar populations have grown over the past 50 years, both in distribution and densities. Since 1971, when they were first declared a big game species, cougar populations have increased in numbers and expanded in distribution in Alberta, which is kind of what you can see on the map in front of you between the left and the right sides. So approximately 2,000 cougars are estimated to exist in Alberta, and populations are highest in the mountains, the foothills, and the southern boreal forest. So these increases have occurred due to changes in landscape use, resulting in more white-tailed deer, which are prey for cougars, and more conservative management regimes, allowing the population to grow. Cougar populations in Alberta are managed with hunting seasons to help maintain healthy populations, reduce conflicts with people and livestock, and manage predation on wild ungulates, and to provide recreational opportunities. So on the right, you'll see a map of Alberta. It's divided into the smaller kind of green units, which are our wildlife management units, and then the orange cougar management areas. So these are units which we manage cougar quotas for hunting cougars. This area is generally where most of the cougar population resides in the province. However, you can find cougars all across the province. So in 2015, Alberta wildlife biologists met with our carnivore specialists to discuss cougar management, specifically cougar harvest and quotas. We agreed that it would be beneficial to collect Alberta-specific data to better inform management decisions around cougars. So the Cougar Adaptive Management project was initiated. So it was a research-based project, and that collaboration, we wanted to collaborate with the University of Alberta on those research initiatives. So we chose 3 CMAs for our study area, shown in the black kind of hatched area on the map. And this is where we targeted our capture work. So that region extends kind of from Whitecourt in the north down to just about Sundre in the south, and then Nordegg in the west, all the way to Drayton Valley and Rocky Mountain House in the east. So these CMAs are made up of foothills and central mixedwood ecoregions, and the landscape kind of varies from agriculture, more agriculture towards the east, and oil and gas, and then forestry in the west, and it spans both public and private land. So the first step in collecting this data was to collar some cougars. So we drove access roads to find a cougar track that crossed the road. Then we used trailing hounds to track and tree the cougar. The capture crew then would hike in to where the location of where the dogs were. Hopefully we find a cougar in a tree. Then we immobilize that cougar with a dart gun. The cat falls asleep, and we climb up to lower the cat down, and then we process the cat. So we sample it, we put a collar on, and then we administer a reversal drug and wait while the cougar recovers. So generally we have about like 30 to 45 minutes once the dart takes effect to when the cat will start to wake up. So during that time, we're constantly monitoring its breathing and heart rate. We mark it with an ear tag and a collar. We take about a dozen morphometric measurements. We collect samples for disease testing. We determine age using tooth recession, tooth wear, and color, and we weigh it. And then we collect some DNA, so hair samples and blood samples. So our project ran from about 2016 to 2022, and during that time, we collared 105 individual cougars. Some of them we collared multiple times over that time span. That broke down into about 35 male, 70 female cougars, and we got over 130,000 GPS locations. So now we have some cougars collared. What can we actually learn from that? Well, using the points that were generated from the collar data, we examined space use, so both home range characteristics and habitat use of cougars by sex, age, and reproductive status. With the movement data, we were able to locate kill sites. So we would walk in and put up trail cameras to learn about predation and interactions with other species. So these photos show cougars with various prey items, including a beaver, which is the image with the video, a lynx in the top right, a coyote in the bottom left, and then their favorite was white-tailed deer. So in this, on this slide, I'll draw your attention to the top left of your screen. So this is an example of what the location data looks like when mom has a new litter of kittens. So kind of right in the middle is a cluster, and that's a kind of a focal point, and you can see how she's moving out and back repeatedly. So once we observe this type of pattern, we would go investigate the site to see if there are any kittens present. So this is a video of a female with 2-week-old kittens. So she's kind of still in the nursery site where she had her kittens. And then the second is a video. So after 4 weeks, we would go back, capture the kittens, and then we'd collect additional data directly from the kittens. So that's the second video is about 4 or 5-week-old kittens. So cougars can breed and produce young at any time of the year, although studies in North America show that births tend to peak between July and September, and they're lowest during like January and February.
Oh.
So Dr. Andrew DeRoche and adjunct professor Dr. Mark Edwards co-supervised our graduate students on this project. So Corey Smaraka looked at space use dynamics and habitat selection of cougars. Kimberly Barrett looked at genetic structure of a large recolonizing carnivore, so the cougars in Alberta and BC. Carter Littlefair looked at biological and environmental correlates of cougar survival. And he also did a paper on chemical immobilization effects of cougar movement. Stephen Andriaszek is looking at predation and scavenging at kill sites. And then Paul is exploring the social stability hypothesis as it relates to human-cougar conflict. And then I am working on finishing my thesis, which is what I'm going to talk to you about for the rest of my presentation today, which is social interactions of cougars in Alberta. So exploring that social ecology of a solitary carnivore. So I'm going to give a bit of cougar ecology around that. Most carnivores are described as solitary and asocial because they don't cooperatively hunt, defend resources, attract mates, or raise their young. So in felids, 35 to 37 species are considered solitary, including the cougar. Following this theory, most cougar interactions are rare and would only occur during the breeding season or like a territorial dispute. So this is consistent with a land tenure hypothesis, which proposes that solitary carnivores regulate that density through territoriality and chemical communication, so sound or scent, rather than direct confrontation. So why does this even matter? Well, social interactions are important aspects of behavior that can further our understanding of species density and distribution. They influence territory establishment, dispersal, and competition. And understanding social dynamics is essential for developing effective management plans, particularly in regions where cougars are recolonizing or there are differences in prey distribution across the landscape. So when I was choosing a topic for my thesis, I read a lot of literature and I came across these papers from the Southern Yellowstone Project. So there was recent research examining the social structure of cougars that suggested they're not as solitary as once thought. So what intrigued me was the new behaviors that Elbrock and Quigley et al. Noted, which was resource sharing at kill sites. So what also stood out to me was the differences between the Southern Yellowstone ecosystem where this research took place and what I understood of our system in Alberta. So a quick overview of the Southern Yellowstone ecosystem. This study took place between 2012 and 2015. Their study area was about 2,300 square kilometers, and their cougar density in the area was about 0.5 adult residents per 100 kilometers square. And a key component of this ecosystem is they have a large prey item, elk, that migrate in the winter. So their findings showed evidence of resource sharing between cougars. So they're feeding at the same kill site. So the majority of these interactions took place during the winter when elk aggregated in the valley, which also aligned with their primary courtship period. So the behaviors were consistent with tolerance and reciprocity rather than cooperation by the individuals, but it was consistent with the resource dispersion hypothesis. So this hypothesis predicts that at higher resource richness, the cost of sharing a territory with others is reduced. So in our system in Alberta, our study area is a little bit different. So the area is much larger at over 31,000 kilometers squared. The primary prey are white-tailed deer, which remain much more dispersed in winter, and the estimated density of cougars is higher at 1.7 adult residents per 100 kilometers squared. So what I wanted to understand was whether cougars in Alberta exhibit resource sharing behaviors similar to those in the Yellowstone ecosystem. So due to the differences between the two ecosystems, I predicted that social behaviors will follow the typical land tenure hypothesis, where direct interactions between cougars will be rare. Males will generally avoid each other. Females will display moderate interaction and primarily with related individuals. And males and females will display interactions for the purpose of mating. So before I get too much further, I just want to introduce you to a couple terms. The first is a dyad. So dyad is going to refer to two individual cougars that have temporal and spatial home range overlap. So they each have their own independent home range, but where they overlap, they're overlapping in both space and time. Second, I just want to define an interaction. So interaction can be classified as an indirect, so individuals that don't meet, but they're aware of one another via scent or sound. So that video that just played was a cougar that was smelling some foliage where potentially another cougar had gone by. And then a dynamic or direct interaction involve spatial and temporal use, and they physically meet. So again, that the second picture where there's two individuals at in that case, it was a kill site. So since I'm most interested in whether cougars are interacting at kill sites, I want to examine those direct or dynamic interactions. So I used a stepwise approach to examine interactions within our study area from home range overlap to identify dynamic interactions, and then classified that by interaction event. So the first step was to identify these static interactions or home range overlap. So I used 99% UD home ranges and selected dyads that showed temporal and spatial overlap. So I visually inspected and plotted the data. I removed dyads that had adjacent home ranges but no overlap of points. I removed dispersing individuals, and I also removed dyads where the overlap was because one of the individuals went on a walkabout. So that, there we go. That narrowed the data set to 104 dyads made up of 58 individual cougars, and that was broken down into 69 female-female, 30 male-female, and just 5 male-male dyads. So using simultaneous fixes, so fixes from their collars that occurred within 4 hours of each other, I determined the distance or proximity that for each simultaneous fix within the dyad, and I pooled them. So the graph on the left shows the frequency of occurrence of those fixes from 0 to 1 kilometer in 100-meter intervals. So you'll notice the majority of the data where cougar A and B are within 1 kilometer actually falls within 100 meters, indicating these points are different from just random encounters. So they were intentionally within that distance, so intentionally meeting each other. The graph on the right, here we go. So the graph on the right shows the frequency of simultaneous fixes occurring between 1 kilometer and 10 kilometer intervals. So this is just showing that there's a greater frequency that the cougars spend farther away from each other. So basically avoiding each other. They spend more time at 9, 10 kilometers than they do within 1 kilometer of each other. So in fact, 0.4 of those simultaneous fixes, 0.4% of all the fixes occurred within 200 meters. So a very, very small percentage of time do they spend within 200 meters. So in order to further examine those interactions, I needed to define the thresholds that I would use to describe what is a direct or dynamic interaction. So I used existing literature by Elbrock and Quigley, where they described a spatial association as a simultaneous fix between individuals that occur within 4 hours and 200 meters. So you may ask, well, how can you really determine what's happening when the frequency of the collar is 7 hours and you're looking at locations that could be up to 4 hours apart? So how do you know what's actually happening? So I asked myself that. So I looked at the data, and due to the programming of collars, 78% of the simultaneous fixes were at the exact same time, and 90% of those were actually within an hour. So there were very few locations that were considered a spatial association by definition, but that were actually 4 hours apart. So I ran an analysis using all 5 of these different interaction indices, which are already in the literature. However, I found that only 3 of them were really helpful in determining and interpreting dynamic interactions between cougar dyads. The next few slides, I'll explain a little bit more about how I used these interaction indices to interpret the data. So here's one example of how I used home range overlap, minimum proximity, and an interaction statistic to help inform these dynamic interactions. So if you look at the dyad on the left, that is a mother and daughter. So it's 2 females that are related. There's 268 simultaneous fixes occurring over a 10-month period. The closest they came together was 954 meters. So if you look at that picture, it looks like the dots are over top of each other. But when you consider the simultaneous fixes, the closest proximity was almost a kilometer away. And so the interaction indicates there was no attraction. If you look at the dyad on the right, it's 2 adult females again. They're not related. the closest they came together was 3 meters, and it comprised 300 simultaneous fixes over 11 months. So very similar data sets. So the dyad on the left was not included in any of my further analysis, as it didn't meet the distance and time threshold for a dynamic interaction. So it kind of demonstrates that you shouldn't assume any dynamic interaction just simply by looking at that home range overlap. So by doing that with every single dyad, I further reduced my data set from 104 dyads that had that static interaction to only 22 that met the criteria that defined a spatial association or a dynamic interaction. So that's only 21% of overlapping dyads. So cougar spatial associations, again, are rare, and it supports that hypothesis that they generally avoid each other. So to look a little closer at those dynamic interactions, I used a definition to identify an interaction event. So an occurrence of spatial association where individuals don't separate more than 200 meters for more than 24 hours. So this clumps some of those points. So you'd get one event if they traveled together for several days and they didn't separate for beyond 200 meters for more than 24 hours. So we're just classifying those as events. So I visually examined each of these events, including a period of time before and after the interaction. I identified if there are any clusters associated with the interactions that could indicate a kill site or a nursery site. And then I animated those dyads to identify whether the movements were cohesive. So I'm going to show you an example of how I use these tools to examine the interaction. So this dyad is a male and a female, and they've got a fair amount of overlap, which you can see in the diagram on the left. The interaction p-value, so in the top right, it's labeled as IAB attract, indicates there's a significant attraction within that 200-meter distance threshold. The line graph that you see on the left is just plotting the distance between the individuals, so the proximity they are from each other over time. So I examined all the spatial associations and I found 2 separate interaction events that occurred within a couple days of each other. The points are circled in red on the line graph, and those are the instances when the spatial associations occurred. So the first event was only one simultaneous fix, and it occurred when the female was on a kill and the male came within 150 meters. The second event was 15 fixes over a 6-day period. So I'll show you that here. So I used the R package wildlife DI to determine the local values of the interaction statistic. The top graph is showing at what point in time there was significant attraction between the dyads. The bottom is showing when the movements between the two are cohesive, meaning they're traveling at the same time in the same direction. So by animating the movement paths, which is what you're seeing on the left, I can verify whether those individuals came into contact and whether they traveled with each other. So I watched the movement pattern of the female, which is the green, indicated by the green dot. And I watched to see if the female, approximately 90 days, which is the gestation period, after the interaction, to see if there was evidence of movement consistent with a mother with kittens. So in this case, 93 days after that interaction that happened, the female localizes for several days and then starts moving out and back to her den site. So that you should have been able to see at the end of that animation period. So I did that process with all of the dyads. I animated them. I looked at all of those interaction statistics. And here's a quick summary of the events I found. So from the 22 dyads that had spatial associations, there were 61 in total events between 24 individuals. And most of those events occurred between females. There was only one event that occurred between two males. So I really won't talk about that much further past this. So here's a closer look at those interaction events and how I classified them. So of the male and female dyads, I was able to confirm 5 of them were mating events. Again, I was able to see them moving together, and then I was able to look at the data and see that behavior of the female with kittens at a den site. So 12 of the events, I could see that the male and female traveled together, but I didn't have the data necessarily to verify that there was a litter. So, but that behavior is still consistent with an attempted mating. So the mating, those 5, the frequency of 5, I was able to confirm. The 12, they traveled together. I wasn't able to confirm a mating. And then 5 occurred where one individual had a kill site. Of the female dyads, 15 were associated with kill sites. However, the duration was pretty low, so less than 2 days. And most of those interaction was a female at a kill site. The second female arrived, pushing the first off the kill. So they weren't sharing the resource. One was taking over the resource. In 10 instances, the females actually traveled together, with several events being over a week. I just want to mention too that we did monitor about 150 kill sites throughout the project using various trail cameras, and we didn't have any instances of adults attending these kill sites at the same time. These are just a little bit more detailed summary of what the data showed. So there were more interactions that occurred in the summer for both dyad types. Females that interacted were more related, particularly those that were traveling together. So to summarize some of these results I found related to interactions between cougars in Alberta. So as predicted, interactions are really rare. The male and female interactions were consistent with breeding events, and those around kill sites did not suggest shared food resources. The female-female interactions were highest related to kill sites and traveling together, but a high percentage of those were explained by relatedness. To wrap up, here's a quick comparative summary of the findings between the Yellowstone and Alberta studies. So the Yellowstone ecosystem is on the left and the Alberta on the right. So even with twice the number of cougars involved in the analysis, the Alberta dyads showed fewer interaction events. The interactions that were associated with kill sites were twice as likely in the Yellowstone system, which is consistent with their resource dispersion hypothesis. And the occurrences of interactions in Alberta were lower, which is consistent with mating or kinship interactions rather than a tolerance for resource sharing, therefore consistent with that land tenure hypothesis. So in conclusion, in high prey or high resource areas, cougars tolerate more overlap and social interaction. In low prey or high competition environments, social tolerance decreases and individuals maintain more exclusive ranges. So Alberta's population follows the land tenure hypothesis, and the social structure of cougars is influenced in part by resource landscapes. So the differences we're seeing between Alberta and Yellowstone is demonstrating that. So cougar sociality is emergent and context dependent. So I just wanted to show a couple of slides to once again thank the individuals who were instrumental in executing the project, including the houndsman, our project wildlife veterinarian, and some project leads and key staff. And even more participants that helped with field work. Many of them were our very important kitten holders. And this is definitely not an exhaustive slide. These are mostly just the folks that I had photos of. So I apologize if you helped out and I didn't include you on this particular slide. Thank you. And thank you very much for listening.
Thank you, Delaney, so much for a very interesting science seminar to talk today. We really appreciate that. Just want to remind folks, our next talk will be on Tuesday, October 28th, where we're going to have Brett Buccal presenting on recovering Alberta's northern leopard frogs, a history of translocations. If anyone is not on our mailing list and wants to be added, please send a message to the email address of the Office of the Chief Scientist. That's epa.ocs@gov.ab.ca. And again, thank you, Delaney, for such an excellent presentation and responding to all those questions. Thanks very much.
Yeah, you bet.
Thank you, everyone.